Hi, everyone. Thank you so much for joining us today for this BioSkryb Genomics webinar. My name is Jenna Gallegos, and I'll be your moderator. In this webinar, Dr. Victor Weigman from BioSkryb is going to present data on using primary template-directed amplification to perform single-cell whole genome sequencing to identify on and off-target events in individual cells that occur following CRISPR-Cas9 gene editing. Without any further delay, I will hand it over to Vic.
Thanks, Jenna. There we go. Share my screen. Excellent. Well, thank you so much for the opportunity here to speak today about what we think here at BioSkryb as a very exciting application of this technology and really helping showcase and exhibit what the power of looking within an individual cell's genome to uncover the underpinnings and the variability of a super rapid growing field in gene editing. And gene editing technologies have not only been on the cover of magazines, both scientific and cultural, but really shaping a nuance of the way that we can have more control over overcoming diseases. And currently in the field today, there's 26 approved gene therapies. And you can see here, and most of those are actual gene therapies. A lot of the others are in the RNA and cell-based therapy. It's important to note, while we'll be talking about CRISPR data today, none of which so far are CRISPR derived. But ramping up to this approval status, there are over 2,000 clinical trials going on, and this is just a survey of what clinical trials exist in what phase in this extremely exciting cell and gene therapy field. One of the things I'll call out for that is the gap or the division here between how much is in preclinical versus that early phase 1. And really, hopefully, some of the discussions that I'll highlight today help identify mechanisms to accelerate that stage gate, going through understanding more about your therapeutic, more about how the cell is behaving to this edit. As far as the use case, the indications these therapies are going on, yes, certainly cancer is a big part of those, but here rare diseases, genetic disorders, certainly in the news, things like sickle cell and beta thalassemia are all being addressed by these therapies, which is really, really exciting. I'll say the numbers we pulled out were from a recent survey by ASGCT, the organization involved in cell and gene therapy. So certainly encourage a look through of that. So how does this work, right? How is this going on? And this cartoon here is really showcasing some of the more common ways this is going on, but is not comprehensive. Right, you have your cells, in this case in an in vitro experiment. You're introducing your vector, which could be anything from a virus to a nuclease editor to liposomes. There's certainly plenty more. Then you wait. Maybe you're applying a stimulus, you're applying a drug, you're just seeing how these cells grow, right? And based on that phenotype, is the cell alive or dead? That may give you what you need to know with regards to your therapy. Or you have a reporter. As soon as it's integrated, you have a floor, very common, that gets lit up to let you know that it's there. Or you're actually running a PCR because you know precisely where your edit is happening, and you're confirming with that very targeted approach what's going on. And these reporters are incredibly important because they're letting you know currently how this stuff works. So these things don't tell you is what's happening inside the cell molecularly, right? To the genome or to the transcriptome, to anything, right? What's the comprehensive view of this? Does your reporter have limited detection, right? Is stuff happening, but you're not able to see it, right? Do you have sufficient evidence with this paradigm to support your mechanism of action? And also, as kind of the title of the presentation suggests, what off-target events are really impacting these phenotypic outcomes, right? At BioSkryb, we actually, as said in the very early introduction, we sell kitted solutions to enable you to identify and amplify these cells that we talked about earlier that just drop into our instrument-free chemistry workflow. So our PTA process, primary template amplification process, thoroughly, robustly, and completely amplifies the genome of a given cell in such a way that it can be sequenced at the end. And following that, we also provide a solution to enable the analysis of that data. The data I'm showing you today went through this base jumper platform that we offer to our customers. So, starting from the very beginning of this process, you can go through your existing research experimental design, but also you can complement by getting a deeper dive into the underpinnings of the genomes following these edits. So, accuracy of this solution we're going to be talking about today. So we've sequenced lots of different controls. We're very fond of those provided by NIST. I have 3 of them here as an example, HG001, 2, and 3. HG001 is known to a lot of people as NA12878. And so we're going to showcase kind of how well that amplification occurs while preserving the base level. So effectively, what we have in this small cartoon to talk about accuracy is we took 10,000 random positions that we knew to be reference across 10 cells from each of these different controls. We expect everything to be blue, which is the genotype of wild type. Gray is heterozygous and red is homozygous. And we have some random positions that we can look at. If we expand this to 10,000 positions, it looks a little something like this with HG1, 2, and 3 over the top in columns. Every column is an individual cell. The rows are the individual positions. You see, for the vast majority of this, it's blue. There might be certain positions that are white. There might be a smattering of pips here where we have single changes in noise. If we look at this in aggregate, not just looking at false positive rate, we look at things like sensitivity and positive predictive value. Again, with single cells that are roughly 25x, which is the data I'm showing you today is roughly in that space. Some is higher. The higher depth you go, the better the sensitivity and the positive predicted value. The lower depth sequencing you do, those numbers retract a little. But if we look at all that and we actually measure all these values here in this larger study, our false positive rate here is certainly less than 1%, which we're really happy with. So again, complete, robust, accurate detection of a human genome feels like this solution really enables investigation of the genetic happenings of a cell or collection of cells as we're doing our experiments. So what I'm sharing with you guys today is research that was actually done as a supplement from the Pena et al. Paper in 2021 in PNAS that really launched the PTA method. I encourage you guys to give that a read if you have not. And certainly the data I'm showing here, you see the core concepts of that in that paper. So what was done is we have 3 different cell types, cell lines, CD34 hematopoietic cord blood cells, U2OS osteosarcoma cells, and ESC embryonic stem cells. With those, they were either treated with a highly specific guide, EMX1, or a less specific guide, VEGF-A. The conditions that we returned sequencing data is bulk sequencing, which we had greater than 10,000 cells, and we did single cell sequencing on the rest, some that were mock transfected, some that had Cas9 without the guide, and the other 2 sets, of course, had the guides I described earlier. What I mentioned is we did 25x whole genome sequencing, and we applied the base jumper bioinformatics platform to do your alignment, your variant calling, your indel calling, copy number, structural variant, the biomarkers that we're going to be discussing at the end of this conversation. Throughout this conversation. So what are our primary questions for this experiment? We want to highlight the edit-specific changes and see how that looks across the different cells, right? We know that cell to cell we have a lot of heterogeneity. What about editing vectors? And then we want to showcase the true difference of guide specificity. When we say that a guide is very specific or non-specific, what does that mean? What's actually happening under the hood? And then we're going to characterize the off-target events. We're going to look at additional indels. We're going to look at copy number changes, structural changes, and we're actually going to add what we would be seeing if we were to leverage prediction tools of off-target events. So let's look at this, the gene here, EMX1 locus. Every row here is a different isoform. The edit we're doing is in this particular exon cartoon right here. Again, the colors are the isoforms. And the pie charts are how often these individual insertions or deletions were observed. Note the size range for these edits can be from 1 base to 50 bases for this particular edit. And sometimes the exact same edit was seen in other cells, but also a variability of those edits. And you can see here the difference in colors here for UTOS and CD34. Those frequencies vary. So if we actually zoom in and look at the changes of these individual cells, so for each of these conditions earlier, the guide treated cells, there are 5 each in each group for each cell line. So these are all CD34 cells I'm showing here. Here's cells 1 through 5. Here you get to see your protospacer and your adjacent molecules. And you get to see here where these changes are. So these edits, are happening somewhere around in here, and you actually get a varied set of indels that are being called at this position. Throughout the presentation, deletions are going to be in blue and insertions are going to be in pink. Okay, well, that's how it looks for CD34. What about the other cells? Well, for U2OS, it's a completely different set of changes in some cases. Some cases, they're complete overlaps, which is great. But now we can really see individual variation. And of course, if we were to do bulk sequencing, some of these things may actually pop up. We can see a lot of commonality here, for instance, for this edit, which is fantastic, right? So let's take the VEGF-A, the area where we knew the guide was not as specific. Smaller gene, less exons. We're looking at the edits here. Okay, well, we did get hits within the area we were expecting, although in a few of the cells, and this is again our CD34 cell line. We're in the area we're supposed to be, but certain cells show signs of having no edits at all. And if we actually go to a different cell line, let's say we're even looking at applying this edit to osteosarcoma, the edit only incorporated in 1 of the cells, so 20% of the cells, which is really interesting. So if we look to characterize the depth of these off-target events, we showcased what on-target looks like, right? We get a lot of variability. We see it across different lines, but even looking back at this CD34, we actually have, and even the UTOS, an off-target event here, a change that's happening close by, not exactly where we intended. So how do we magnify that? So we looked at positions that are between 5 to 50 bases. We saw them also in the 75 and 100 base pair area, which is outstanding. So if we actually start counting those, this is for the CD34 as a point of an example, and these are our more specific EMX1 edits. We actually see that these off targets are happening in the intergenic where we expect. I think most publications cite that this is where these things are happening. We also see it in intronic areas, but really we see a lot more in exonic than we were expecting. And if we were to leverage we, in this case, used Cas9 Finder in this particular approach to predict locations of off-target events. We see that, and that's in the blue here, we see that actually, there's a vast majority of the off-target events that are missed through bioinformatic prediction alone, which is, not unsurprising, but I think the magnitude itself is fairly surprising, right? We know the genome, we know the sequence, we can kind of understand if we make some changes, single base, multiple bases, we can use the reference to get a pretty good estimate of where these things should go. But biology, as we all know, can be a little bit more complicated than our initial models. We do know there's more prediction algorithms, but for the purpose of this presentation, we leverage this one, right? So moving on there, what's happening underneath is a lot of variability. Again, I split the U2OS and the CD34s here, but you see, and the light blue is in the het, the dark blue is in homozygous. What we're able to see, certainly with our technology, is the zygosity of changes. So you get to know that, hey, we had an edit of 1 allele. We had an edit of both alleles, which is fantastic. We see also the variability of different sizes. And we also capture in this particular plot which ones were called by bioinformatic tools. So again, we see a lot of changes here. You see it's being variable. And of course, for some of these positions, they might have been seen in only one of the cells in our group, right? 15% of all these positions here, and there's way more that just truncated for viewing. Um, if we have that low, how many of these are we potentially missing by looking at traditional bulk sequencing, right? Obviously, quality of guide, of course, may impact how well the on and off target goes, but I think the magnitude here, even for our more specific guide, is worth noting and investigating further. So, copy number changes. So, while these treatments are meant to do small editing, copy changes in the insertions and deletions of a given region we want, we're actually seeing the impact of that off target isn't just more insertions and deletions, but we see changes in copy number across the entire cell. So what we have here is the 3 cell lines I talked about earlier, the CD34, the hematopoietic cord blood. Even our more specific EMX1, we still saw, in this case, a change in chromosome 18 and in 21, where VEGF-A maybe only had a change in 13. So at least they're getting to different areas. I'll point out that part of having the bulk control and individual cells that are untreated, we use those to do somatic mutation calling. So we corrected for insertions and deletions and copy changes and structural variants that were not in the vehicle or the bulk controls. So we're trying to represent here what's happening in the individual cells that are directly related to the guide and the Cas9 being introduced into the system. So, U2OS and osteosarcoma cell lines certainly would have expected aneuploidy. The vast majority of those were corrected out, and we still see guide-specific changes in there, and certainly a lot more than we did in the CD34. One of the most telling parts of this is in our embryonic stem cell, we saw 4 different events comprising 153 megabases of sequence, only happening in our leaky or our less specific edit. So again, even though we don't expect aneuploidy changes in these embryonic stem cells, I think it's an even better tell of what potential outcome can happen with these editing technologies. To kind of like a blank slate cell. So editing impacts to structural variation. So we are creating insertions and deletions, but in addition to, again, these indels, these copy number changes, we're actually seeing translocations happen in these cell lines and other structural variants. Again, we're coloring the lines here by EMX1 or VEGF-A, the guides that we're leveraging here. But we do see, in this case, you can count that we've got 5 different translocations in this cell line from these edits. Same thing for the U2S cancer cell line. We see 3 very striking one. The embryonic stem cell line, just, wherever it was void of copy number, it seems that in the translocation aspect, much stronger, even in the other 2 cell lines. Again, while primarily we saw earlier being driven by this less specific guide, we do indeed see a translocation coming from our more specific guide. So again, not only the off-target indels are something to consider in that variability cell to cell, these structural events, these copy number items, these structural variances, the translocations are also occurring in the presence of these editors. And I'll call out that to make it into the graph here, we had to make sure your translocations are happening in at least 2 of the 5 cells. We did have locations in here that occurred in all of the cells, which is maybe 1 or 2 specific for each cell type, but it is striking then it's kind of highlighting the variability between the cells of things that are occurring in response to this editing structure. So for this, smaller experiment, the primary questions we had, we're able to get a lot more clarity on. So, understanding the cell-to-cell heterogeneity, especially in the presence of edit? Absolutely, right? We saw that the types of edits, right? When we zoomed into a given region, we saw that the insertion and deletion types changed by cell, right? Few events happen uniformly across cells. And I think this is a caution to leveraging bulk sequencing alone as a mechanism for identifying impact of edits, because these things may be missed if you didn't sequence deep enough, or if the event in question is exceedingly rare. So we show the variability of guide incorporation, right? We saw for specific guides, which is fantastic, we saw changes in each and every cell. For our less specific guide, we saw changes in 20, 60% of cells. And commensurate with that, the off-target amounts changed in scale from these editing vehicles. Looking at off-target impacts, there certainly the off-target events are happening more commonly than we previously knew and understood. The prediction tools that one may use, at least certainly the one that we leveraged here, may only capture 15 to 20% of real events. And I'll also add that in addition to indels, which most people understand as what the editing is doing in the first place, having those in regions that are unintended, especially the volume and magnitude of those, are showing almost directly correlated to having changes in copy numbers of parts of the genome, as well as getting structural variants such as translocations. So we showcased in a very kind of brief experiment what happens when we're looking at changes in the genome. But what about changes in the genome and the RNA of the same cell? As I mentioned earlier, BioSkryb provides kitted solutions that enable you to not only look at the amplified genome of a given cell, but the genome and the transcriptome of the same cell. So we know because of adding the RNA part of the cell is important. That's certainly how a lot of single cell sequencing is being done now. Resolveome specifically is not just providing gene expression, which is important. There's isoform level expression as well. We're able to see, because we're amplifying up the individual transcripts, that we're able to see a broader detection of isoforms. We're also, again, because of that amplification of the isoform in the cell, we're seeing a much broader gene body coverage than a standard droplet based sequencing technology. Again, the resolveome is in blue and droplet RNA-seq is in orange. Even more interesting than that is we're seeing here, every row here is an individual cell. Every column is a transcript. We ranked the transcript by their length, and we see that resolveome is able to detect much longer isoforms than your standard droplet sequencing. So we're bringing a highly resolved transcriptional product on top of this well-characterized and robust genome amplification method. And because of those impacts, yes, the standard outputs that most people expect with this can be done, right? We have cell ID, which can be used to see the collection of cells and various functions in your tissue of interest, but we're also getting cell cycle state. And that's an important thing to note on as well, because it's certainly published that success or potential interacting features of genome editing can be affected by cell cycle state and how much of a given part of the genome is actually available. We could do variant calling, again, a larger genome body, differential isoforms, longer isoforms. We could do a lot more variant calling on a given transcript and across the transcriptome than, again, a standard droplet-based sequencing. And I talked already about isoform detection. Blending these two together, allele-specific expression certainly is part and parcel to having a genetic event that leads to a transcriptomic event where you're shifting expression of not just an isoform, but a given allele in that isoform, which has the most potential to have real protein change. So we see Resolveome as a complement to a lot of current testing methods that people are already using to understand the functionality of editing methods. So Perturb-seq, and I have just one of the papers of the many that here in Perturb-seq with Adamson et al. In 2016, the cell. What we're able to empower with Resolveome without spending the whole talk going over every type of functional testing method, beyond what the existing Perturb-seq can do, Resolveome empowers richer expression. So you can understand more features of what pipeline pathways are being expressed. You're, again, complementing that because we added that DNA. You're actually getting the genomic location of the guides or the specific changes that you're getting. And of course, even if you're not doing a base editing, but doing a viral or extracellular chromosome or things like that, you're getting the genome of this without targeting. I mentioned earlier about allele-specific expression. So you get this, the idea of the perturbation that you perceived, you've confirmed it, and you're getting the expression, you confirmed it in the DNA, and you're getting the expression in the RNA to say that it's having the intended effect that you wanted. And Discover-seq, you know, we're able to, we believe we can do unbiased, again, genome sequencing has our ability to do a broad set of on and off target events, which we showcased earlier in the presentation. Plus, we have an unbiased reporting of loci, right? If you're performing whole genome sequencing, certainly we're capturing everything that has events that weren't seen in our mock controls. For GuideSeq, we can further qualify the variability of changes due to the edit. And we're less affected by changes in copy number that may thwart getting a fuller picture of where the editing items are happening. And for Site and CircleSeq, certainly with the genome and transcriptome unbiased workflow, we're a little bit less complex as far as the mechanical steps in SiteSeq. And CircleSeq, you know, we have less restrictions to predict cleavable guide sequences because we're sequencing all the way through versus cleaving prior to detection. And of course, the expression can really confirm the impact of those changes that you're assaying in a broad spectra here. So again, all these fantastic functional technologies to understand the impact of edit. You know, Resolveome is just adding to those with that genomic place for areas that didn't have that, but also when we're looking at both, we're able to see the intended transcriptional impact as well in an unbiased approach, or leave the researcher with libraries that can be captured in ways that they prefer. So take-home messages here is, you know, Resolve DNA is robust and highly accurate. I showed at the very beginning our sensitivity, our positive predictive value, our false positive rate were all very good. So we feel confident in Resolve DNA's ability to amplify the genome of a single cell. Also, single cell sequencing offers resolution to identify how frequent edits can be missed or misrepresented in bulk sequencing, right? So if it doesn't happen in a bunch of cells, you're going to be harder pressed to find it in standard traditional bulk sequencing. We're gaining sensitivity for on-target edits, as well as getting incorporation frequency. So not just an overall variant allele frequency, but a cellular frequency in addition to within cell zygosity measures, right? Is your edit affecting one or both copies of the genome? We also notice that off-target events are happening at a higher frequency than previously understood or reported by bioinformatics tools. The impacts of these edits are happening beyond indels, right? They're impacting larger swaths of the genome, from copy number to structural variants that current assays or functional tests are likely going to miss altogether. Being able to report both the DNA and RNA of a single cell offers a really unique opportunity to match the edits that you're making to a functional impact, which is really awesome. And so kind of summarizing what I've showed in our CRISPR experiment here is, and the pie chart represents all possible edits that could be happening within the cell. In the orange here, we do have a lot of these are being found by predictive tools, which I did mention earlier, but the sensitivity of prediction tools to call off-target edits that we observed was very, very high. So it may not have captured everything, but what it did capture molecularly, we confirmed. So this is certainly a subset. The vast majority of the events happening in off-target are insertions and deletions of, again, that kind of 1 to 70 base size that was totally missed previously. Also, we get a little less than 1% both of copy number changes and structural variants that were happening in the DNA of these affected cells. And kind of moving beyond that, adding that RNA layer, right? It's unknown what potentially we're missing from looking at things like isoform detection, allele-specific expression, fusions, pathways, right? We have a really great solution here to blending together DNA and RNA with long or ability to detect isoform transcripts. It'd be interesting to see how we can complement what we're already learning about genetic changes to impacts to RNA and the functional changes. So we're really excited about hopefully coming back to you guys again with the impact of the RNA. So we'd like to posit a kind of thought experiment to the audience here, although this is not isolated to this based approach, but in an effort to help accelerate the path, the discovery path, making sure that you reduce your time and overall cost trying to figure out how a therapy works, right? We highlighted at the beginning the variability and the sheer magnitude difference of the preclinical to the phase 1 testing and clinical trials. Hopefully a mechanism like we're talking and showing here may help accelerate this, right? We want to characterize the parental cells, right? This is happening in bulk sequencing already. But if the cells or the system you're working on has highly heterogeneous calls, you might want to know that. You might want to know that there are clonal populations in your studied system that may impact the efficacy of your edit, right? You're doing your edits, you're doing expansion and clonal population shifts, outstanding. So as we actually start screening for on-target editing and copy numbers, you know, we can look at resolved DNA, which is even at a low pass, not a full strength whole genome sequencing, the number of candidates that we get to see in this step is really, really high. And the cost is low in this case because it's less cells that we're looking at and maybe at a lower strength. We can leverage what we did at the beginning of this cycle, typing the control or system, so DNA, to allow us to further correct the edits we're finding here. Then, of course, looking at really attacking the sequencing cost and exome or panel sequencing plus phenotyping. We talked again earlier about these reporters. Here we're seeing all of it together. We have the reporter, we have the targeted edits and off-target edits all characterized together. Now we can start doing a little bit deeper sequencing. We probably have to sequence a little less cells now as well, because we can be guided by the phenotyping and the reporting to specifically select for cells we're after, right? So again, as you're going through this process, you're increasing the resolution, right? You're decreasing the total number of things you're testing, but you're increasing what you know about each of those, to where now when you've got some solid candidates, you can do a full strength whole genome sequencing. Right. At that point, you're doing it on way less cells. Your cost is going to be higher because whole genome sequencing is still really expensive. But now you're really characterizing the inner workings of edits that have made it this far down the pipeline. until you get to the point where you're selecting that final candidate. We'd love to help any and all of you with this process. We understand the value, the importance, and the, complete changing experience this therapeutic opportunity presents, which is why we're doing this talk. So again, thanks. And just real more, we would love to talk to more people about cell and gene therapy. You have some more opportunities to chat with BioSkryb and our awesome staff coming up. We will be at ASHG. Dr. Asai Salas-Gonzalez will be talking about more of this data in deep that I showed you today. At our poster session there or at the booth. We can certainly arrange a discussion if that is of keen interest to yourself. I also did a bunch of the analyses for this platform. So what you saw today was precursors to the visualizations we'll be adding into the Base Jumper platform. Also, as we've advertised before, BioSkryb is actually part of the NIST Genome Editing Consortium. And they're going to be having a workshop on the first 3 days of November of the measurements and standards for advanced therapy. So if you're really interested in this kind of stuff, come to see what all the other experts are going to be talking about. I will be at the conference on the right. Love to talk and meet with more of you. With that, I'll conclude. We do have on our website ebooks and tech notes outlining applications of cell and gene therapy. We've got a QR code for our website, and I'm happy to take any questions that you guys have.
Awesome. Thanks so much, Victor. We've got a ton of great questions, so we'll try to get through them as efficiently as possible. Starting with just some quick technical questions. How many cells did you analyze for each cell line, and what sequencing depth was used?
Sure. We did whole genome sequencing for all the cells. It was 25x. For the each of the bulk controls and the mock transfected and Cas9 only cells, as well as the individual treated cells, we did 5 cells each. So across 3 cell lines.
And in terms of your results, what was the percentage of each on-target versus off-target event for each mutation category? And do you think you reached saturation or captured all of the events?
I do feel like we captured, because we really looked over this data with a fine-tooth comb. So, with the on target that I showed in the plots earlier, we're capturing, even though I showed 1, because I can only show 1 axis, there's probably on target across those 5 cells that we looked at for each guide. There's probably about 10, 12 that are happening right on target. Even the same cell might be edited multiple times, which we do see. If you open up the genome browser, you can see it. So we've got 10 in one hand for the gene of interest, for the cell type of interest, and the off target, like I showed with the CD34, was something like 650 in the introns alone. So that's including the intergenic regions and the exomes. You stack all those up together, that's roughly about 1,400. So you've got 10 over 1,400. So we're really, the on and off target ratio is pretty vast.
Awesome. Thank you. Really interesting question. So considering that we're using cancer cell lines and they may be experiencing genomic instability, how do you discriminate between spontaneous and CRISPR-induced translocations?
Yeah, that would be great. That's why we added at the very beginning of the controls the bulk version of these cell lines, as well as individual cells that were both mock transfected and without the guide. So we used mutations or variants or alleles that were found in each of these to subtract out from there, including, as I mentioned earlier, the structural variants and the copy number. So while it is not perfect, it's as good as the technology allows, so that what I'm representing in that cancer cell line is already taking the fact of it being a cell line and the variability of the molecular events. We're taking that out as much as we possibly can. And if you saw especially for the copy number plot specifically, the number of copy changes was super small. It was, maybe 10 super tiny ones. Whereas if you looked at that cell line in the bulk sequencing, you're going to see gains and losses across the entire genome. So we feel like we did a really robust job of pulling out those cell line specific changes.
Very good. Thank you. Can you comment a little bit on the Cas variants used and, maybe expand a little on what we'd expect to see with different Cas variants?
Yeah, so with again, this is a kind of toy first pass example. The EMX1 was chosen as known to be a fairly and published specific edit a guide to say like, hey, we're going to make changes in this one exon. I showed the one exon there, and we did. We hit every single cell that we expected. So the edits are working. They're hitting the cells that they're wanting to. Awesome. Right. So we know that it was supposed to be specific, and it was. And the off-target events for that, which I mentioned earlier, even for our CD34, the hematopoietic stem cell, or yeah, cord blood rather, it's the off-targets were still in like the over 1,000. Right. Whereas, and I didn't show this because I didn't want to overwhelm the audience with too much data, but when you start looking at things like the VEGF-A, the less specific editor, um, not only was its incorporation at 20 and 40% of the cells, independent of the cell lines, its off target for copy numbers and translocations was double that of a specific one, and its off target indels was also vastly more than the precise one. And so we chose these as a kind of, we know the truth is lying somewhere in the middle, or even with the newer guide technologies, even better than the EMX1. But we wanted to showcase the polls to allow the audience to see that maybe their research is somewhere in the middle of the spectra.
Gotcha. Thanks for that. Let's talk a little bit about the predictive algorithms. What all algorithms did you test against your data? And for Cas9 Finder, what mismatch or bulge thresholds did you use? And did you dig into the off targets that were missed? Can you theorize a little bit about why they might have been missed?
Absolutely, yeah. So for this, we did leverage the tool Cas9 Finder. It's been around for a while. Since running this experiment, we found 3 other ones that we're looking to try. It won't be at the poster in 2 weeks, but how you choose those, it becomes a little bit more of more than just the searching of making the chains and aligning on the genome. But we allowed here 7 potential mismatch events for every 15 base protogene, which is a lot. That's almost half, right? So we allowed a lot of variability. Right. So just to assume the algorithm is having the best possible chance at finding things, right? Insertion here, deletion here, across all the other methods. But again, that's a straight lookup. As algorithms get more and more sophisticated, understanding the processing, the temperatures, the context specific, so the alleles in a certain order change, are going to impact a lot of these things. So again, the improvements, just like the guides, the molecular improvement is being there. I think these algorithms are getting a lot better. I'm certainly eager to use them. But again, for this, we saw, several hundreds that AllFinder was identifying with that 7 base pair flexibility.
So when we're talking about off-target effects, obviously everybody's mind is going to go to regulatory. Is BioSkryb able to offer any kind of regulatory support, or is there a way to use the platform to generate data that would go into IND?
That's a great question. I mean, the BioSkryb kit solutions are RUO, right? So while we do process those kits in a controlled facility, that's up to an individual lab or a CRO to help build an LDT-based process that suits your needs. Right. Same thing for BaseJumper. BaseJumper is an RUO-based analysis platform. We'd love to help and be involved in that process. But yeah, that will be something. Finding the right lab, we can help provide the materials and ingredients to do that. But yes, not this time. This is a research area. It's meant for exploratory studies at the current moment.
So this next question takes us back to the fundamental PTA technology. Of course, most off-target characterization is sort of thwarted by a poor signal-to-noise ratio. Can you talk about how PTA achieves, less amplification bias and improves that signal-to-noise ratio?
Absolutely. So as just to be super brief here, so as we kind of open the cell in order to allow access to the DNA, the randomness of the starting primers of this are chosen, of course, certainly as really random, but as the amplification is happening, this workflow is also on our website, and I recommend people to look, as we have some super nice videos there to check out. But as your amplification is happening, you have a terminator that's coming on top of those. So therefore, when a polymerase is trying to do again another round of amplification, that's what PCR is doing, right? You have one that gives rise to two, that gives rise to four in traditional PCR, and here that termination process drives the polymerase back to that primary strand, along with the randomness of the starting spots. So one thing I didn't talk about is not only is that randomness happening across an entire genome, it's happening across both copies. So I didn't talk in great detail in the presentation about our allele balance, but we're pulling those alleles from both copies of the genome. So it is, very random, very robust, high coverage because of that terminating process of the subsequent sequences, which are long, roughly on average about 1,200 bases. It's true for our DNA and RNA. It allows you to get a big piece of genome before you're doing your sequencing. So I think because of that, we're extending longer than I think a lot of other amplification technologies and sequencing workflows are going to give you.
Awesome. Thanks, Beck. Next question, are you using, or did you use any kind of pre-selection process to choose or sort the cells that went into this analysis, maybe based on their phenotype?
Totally random. Yeah, totally random cells. As I mentioned in the kind of potential detail earlier, we could use reporters. We could use things like flow cytometry to do that selection going forward, but this was just random selection of cells. But we do find a lot of power in using those reporter and phenotype mechanisms to do your cell selection. So you're having to do less cells randomly.
Gotcha. So we're talking a lot about single cell. Do you recommend the approach you presented with combining single cell and bulk sequencing to verify single cell derived CRISPR edited populations? Or would standard bulk be enough, considering monoclonality of the single cell derived clone?
That's a great question. So a couple different ways to answer that. So number one, yeah, bulk sequencing. If you're working in a cell line based in vitro, you're working in organelles, you're going to get you're going to identify the major events happening in that edit. I mean, you just are. That's just the statistics of the genome copies you're measuring. But what I showed in, and hopefully a bunch of different figures here, for these off-target events that are happening in maybe 15, 20% of the cells, if those are hit, then they're going to be happening in 7 to 10% of the cells. You're starting to hit the noise level that you're going to get with bulk sequencing. And if you've sequenced your model system, you understand where these edits are happening, both on or maybe off target if you're doing a not targeted approach. Awesome. But if you're getting a phenotype or reporting structures that you don't understand or expect, this is where the reflex action of looking at single cells really gets you. And also, if you're really understanding more or needing to really flush out that mechanism of action, this is why I talked about the resolve-on process. You're getting the genome and the transcriptome within a cell. So you would be able to leverage the multiple cells per tissue to independently control or confirm the mechanism of action you're predicting, which again, a bulk, which is again suitable for most things, are averages, but it doesn't necessarily confirm that those things are happening within the same population of cells. What we're showing here is that you can confirm that by doing it in the individual cells first, or as a complement to that original bulk approach.
So that explains why the single cell approach is important or maybe essential. Can you talk a little bit about how essential whole genome sequencing is? If we had done sampling of mutation hotspots or something like that, do you think we would have missed a fair amount of those off-target effects?
You would have certainly missed a whole bunch of them, right? And the question is, it really depends on your research, your platform, your guide, right? I'm certainly not one to recommend everybody is spending more in whole genome sequencing. However, at fractional cost, getting incomplete pictures is going to take the wind out of your sails very quickly if you're not getting the intended effects as you progress down the pipeline. This is why with the thought experiment that I showed a little bit earlier is like, okay, we're going to screen a little bit at low pass sequencing. Maybe we use the RNA to concern the functional aspect. But if you're really trying to project, confirm, and refute your mechanism of action of that edit, I think understanding the whole genome picture is a vital and necessary step to really rest at ease that we cover the whole genome, we're qualifying everything that we can, we're pointing back to the functional events, and as far as science allows, we're doing our due diligence to ensure the safety of potential individuals on these edits.
All right, one last question. Is this data set going to be made publicly available?
We are working on that. So my intent, I would love to make it public. It was a supplementary data set. So stay tuned. If you're already on the webinar and you're getting the notes, I'll be happy to send that out when we post that data. So yes.
Well, thank you so much, Victor, and thank you, attendees, for your very engaging questions. If you would like to learn more, you can do so at bioskryb.com or by reaching out directly at info@bioskryb.com. Thank you so much, everybody. Have a great day.
Cheers.